Okay, so now we're going to head into the third segment in this series overviewing uh, immunology and immune responsiveness. In this third part, we're going to finish up some of the discussion that we started in the previous segment, talking about adaptive immunity. The last time we were discussing this, we were mostly concerned with humoral immunity, antibody-mediated immunity, but we're going to finish up with a discussion of adaptive immunity in the context of cell-mediated immunity for this segment. In addition to that, we'll follow up a little bit with a few examples of how the immune system can go wrong. Without the immune system, we would have a really hard time protecting ourselves from infection and malignancy and all kinds of things that are good, but there are a few opportunities and actually diseases that can be caused by the immune system. So we want to at least make mention of those, even though we really don't have time today to discuss them in any detail. Okay, so let's move into this discussion by pointing out that there is another arm of the immune system. We refer to it as CMI, or cell-mediated immunity. Once again, there's a division of labor in this arm of the immune system, as there is in just about every arm of the immune system. Lymphocytes all working together for a common end, which is to reject a potential microbial infection. In this particular form of immunity, though, that lacks antibody, there is not a B cell in sight. So one of the main features of cell-mediated immunity is there's no B cell response, there's no antibody response. But there are multiple cell types that are involved in the immune response during CMI, and they're listed here. The main player is a T lymphocyte rather than a B lymphocyte. These T lymphocytes are capable primarily of killing things, of killing other cells, killing other cells of the host, sometimes killing other cells of the infection, the microbial cells. There are all kinds of ways that these T lymphocytes can kill other cell types in order to affect protection against an infection. And we'll talk about a few of those mechanisms in a minute. The second type of T lymphocyte, a second subset of T lymphocytes, actually, don't kill anything. They're regulators. They regulate the killers. They upregulate, enhance the ability of the killers to kill. They sort of serve the same function as the helper T cells served in enhancing the activities of B cells during a humoral immune response. So these two subsets of T cells work hand in hand to get the killers up and running. And then finally, the third subset of cells in this division of labor is once again our dendritic cells. They are cells that will ingest and digest and then present antigen fragments to the T lymphocytes. So they're sometimes referred to as antigen presenting cells, APCs. And these APCs are absolutely required for activation of T cells, any T cells. T cells cannot be activated by antigen unless the antigen is presented on the surface of an antigen presenting cell, such as a dendritic cell. So these three cell types work together to generate a population of killers that will take care of the business of rejecting an infectious pathogen. Okay, so in CMI, in cell-mediated immunity, as I said, the T cells that are generated are capable of killing things. So killing can be a process that directly eliminates the pathogen itself by physical contact. And by the way, that's a characteristic of cell-mediated immunity, which is that the cells, the cells that are creating the immunity, that are rejecting the pathogen, they actually have to go somewhere to do that. They have to go where the pathogen is, which is why we call it cell-mediated immunity. In humoral immunity, in antibody-mediated immunity, you noticed that the B cells get activated, turn into plasma cells, and then the plasma cells squirt out antibody, that soluble protein that goes into the circulation and hunts down and destroys the pathogen. The B cells don't have to go anywhere. In cell-mediated immunity, though, these T cells, these killer T cells, have to go places. They have to go to wherever the infection is. So anyway, so one form of CMI involves the killing of pathogens in an indirect sort of way. And the way that they can do that is by going to the site of the infection and secreting what we refer to as pro-inflammatory or inflammatory cytokines. These cytokines are sort of hormone-like molecules. They're small molecular weight proteins that are secreted by one cell type that then will activate the activities of another cell type, sort of like a hormone might do. But they're not hormones. Don't let me confuse you there. Cytokines are not hormones. They're cytokines, but they're signaling molecules basically. They're produced by one cell type to activate the activities of another cell type. And some are particularly good at binding to and triggering the activities of cells of the innate immune system, like macrophages, for instance. So these pro-inflammatory cytokines can activate inflammatory cells, thereby enhancing their activities. So if you remember this really poor quality image that I showed you some time ago, these are macrophages that have in their cytoplasm, you can see here these little dots. The big blob is the nucleus of the macrophage, and if you have a really good imagination, you can see the outer membrane of the macrophage right here. Here's another one over here, so forth. But in the cytoplasm of the macrophage are these little dots that are parasites that the macrophage has ingested. And so without any further activity by that macrophage, those parasites would just live there happily being protected within the confines of that macrophage's cytoplasm. We need to digest them now because we know phagocytosis begins with ingestion, which has already occurred, but then digestion has to then subsequently occur in order to finish the job. In the case of macrophages, they have a very low level ability to digest whatever they ingest. I mean, they can, after a long period of time, a lot of these parasitic cells would probably be digested, but very slowly. And as the parasite multiplies, replicates itself within the macrophage, you'd end up in a situation where you'd never catch up with them and the parasite would win. So what we need is something that will accelerate the digestive activities of the macrophage. And so what we can do is we can provide them with a signal, a trigger that will get them more aggressive about it, get them producing all the digestive mediators within the cell that will digest the parasite much more effectively. One of those mediators is listed here in the legend, which is a cytokine, an inflammatory cytokine called interferon gamma. It's one of the interferons. There are many different kinds of interferons. Some interferons are antiviral proteins and so forth, but this has often been called immune interferon. Its main job is to signal cells of the innate immune system, particularly macrophages. So one thing that I hope that you'll carry away from this lesson is that interferon gamma is an extremely potent activator of macrophages, extremely potent activator of macrophages. And the interferon gamma is produced by T cells, the T cells of the cell-mediated immune response, which have been activated and have been attracted to the area in which an infection is going on, which is where these macrophages are, trying desperately to clear out the infection on their own, not doing a very good job of it at first. But then the T cells get there with their interferon gamma, squirt out some interferon gamma, and then stimulate those macrophages to increase their digestive activities. And as you can see in the bottom panel here, now the parasites are just about all gone once the cells have been treated or exposed to interferon gamma. So that's one way CMI can work. It's simply by providing inflammatory cytokines that will help clear out the infection by cells of the innate immune system. A second type of killing that we observe is killing of the cells that might harbor a pathogen. So we mentioned before the fact that some pathogens have learned how to live inside of host cells. This is particularly the case with viruses. Viruses, as we've labeled them before, are obligate parasites. They can't replicate. They can't do anything when they're outside of host cells. They have to get inside of host cells. Well, once they're inside of host cells, it makes it really hard to get antibody after them because the antibody can't get into the host cells. So what are we going to do about it? We can't get any of the mechanisms or the players that are trying to destroy the virus inside the cytoplasm of a host cell. So what we do instead is it's kind of a dramatic way to end the virus, which is to end the life of the host cell that they're living in. So if the virus can't survive outside the host cell, kill the host cell, and now the virus is outside the host cell and the virus is destroyed. So killing of host cells in which the pathogen is hiding is a major mechanism for clearing viral infections. The cells that do that killing in this fashion are often referred to as cytotoxic T lymphocytes. So these are the same sorts of effector cells that we were talking about before, killer cells, but in this particular scenario, people give them the label cytotoxic T lymphocytes, or CTLs. So CTLs have a very prominent function, which is to kill host cells that viruses live in. And just to give you a little bit of a visual opportunity to see how this works, this is a scanning electron micrographs, a series of scanning electron micrographs showing the cells that are participating in this scenario. So what we see here is this big old cell down here that has a virus in it. So this is a virally infected host cell. And one of the things that happens to a virally infected host cell is that there are certain surface changes that occur. So certain things happen to the membrane of the cells that harbor these viruses. And those changes can be recognized by particular subset of T cells, the so-called CTL, the cytotoxic T lymphocyte. And here's one right over here. So we have two cells right next to each other. Here's a big old fat host cell. I don't know what kind of cell it is, but it's a host cell that's infected with virus. And here's a T cell that has recognized certain surface changes associated with the host cell, indicating that there's a virus inside. So this CTL is specific for those very specific changes that occur that are associated with that particular virus. And so they bind. So that's the initiation of the process that's ultimately going to eliminate the host cell that's got the virus in it. In this second image, what we notice is that there still are the same two cells there, except here's the CTL over here, and here's the host cell. Now, if you notice, if you compare the host cell in this image compared to the same host cell in this other image, you'll see that there's a dramatic difference in the surface, the way the surface of those cells look. This is very telling because what we see is that in the original host cell, there are a lot of it's a very dynamic membrane with a lot of membranous projections kind of sticking out, little finger-like projections that stick out of the host cell membrane. And that's a sign that host cell is pretty happy, actually. It's very healthy. It's making lots of energy. It's got a great nutrient supply. It's doing whatever it wants to do pretty much. And those projections that come out, sometimes the cell biologists will call them membrane blebs. They bleb. So this host cell is blebbing like crazy. It's really happy. And usually on the points of these finger-like projections will be, transport proteins for nutrients or adhesion molecules for sticking onto surfaces or what have you. Anyway, it's a very happy blebbing cell. In the second panel, though, you can see that all the blebs have pretty much disappeared. And that's because it costs the cell a lot of energy to create these blebs, these membrane projections. But this cell right now, after just a few minutes of being bound by the CTL, is in big time trouble. It is beginning to degrade. It's beginning to get damaged. And so it is desperately right now diverting energy to repairing its own physical status, repairing its membrane and repairing other things. And then as you wait a few more minutes, what we see is that in this third panel, the CTL is gone. So this CTL over here just kind of releases itself once the target cell has been damaged sufficiently, and it goes off to find another victim, another virally infected target cell, host cell. And then it'll begin the process all over again. Meanwhile, the original target cell has been damaged so severely, it's gone down a pathway of programmed cell death, actually, that leads to the disintegration of its DNA and disruption of its mitochondria, so it can't make energy anymore, and its outer membrane is beginning to deteriorate. It is beyond the point of no return, and it's dying a rapid death until finally, in this fourth panel, you can see this same cell. This is an amazing photo that was captured, but when whoever pulled the trigger on the shutter of the camera really lucked out, because this is a cell that's right about to burst, lyse. It's porous, and it's just about to disintegrate. A split femtopicosecond later, it would be gone. So this is a really an amazing photo right here. This programmed cell death that I'm referring to has a fancy name, which is apoptosis. And I don't know if you've studied apoptosis in any of your other classes, but it's a way of triggering death in certain cells that you need to eliminate for a variety of reasons. This is one reason, getting rid of a virally infected host cell, but there are a lot of reasons that you might want to get rid of certain cell types in the body, and they usually do that by going into this programmed cell death sort of pathway. Okay, so just to kind of put together all the different cells and mechanisms by which CMI can work, antigen comes into the body. It might be a virus, it might be other things as well. Triggers T lymphocytes, a particular subset of T lymphocytes. Not all T lymphocytes can do this. Some, remember, are those T lymphocytes that help B cells, and there are T lymphocytes that do other things as well, not CMI. So there's a whole bunch of different subsets of T cells that we're not going to even mention today, but it's very complex and very elaborate, and with regard to all the different functions they can participate in. But in any event, in this particular case, one particular subset of T lymphocytes, which we sometimes refer to as inflammatory T cells, are those cells that are able to make inflammatory cytokines. So these are the T cells that would make, for instance, interferon gamma, like the example that we used a few minutes ago. There are other kinds of inflammatory cytokines that we won't get into now, but one of the most major inflammatory cytokines is interferon gamma, and the main purpose of that is, I'll get back to that in a second. The main purpose of that interferon gamma is to enhance the activities of phagocytes, like macrophages, like neutrophils. So it enhances activities of cells of the innate immune system. This little molecule that just bounced in here is CD4. CD4 is a molecule that is common to the inflammatory T cells. And for that reason, it serves as a great marker if we want to monitor where they are and what they're doing and so forth. It also happens to be the molecule that the human immunodeficiency virus targets when it kills cells of the immune system. So CD4 is the receptor for HIV. just another little side thing. Okay, meanwhile, we have the antigen that could possibly trigger another subset, a different subset of T lymphocytes, but T lymphocytes that participate in CMI in a different way. The main example that we gave was activating the subpopulation of T cells that we referred to as CTLs, as cytotoxic T lymphocytes. And the main thing that these cells can do is to kill other cells. That's why we call them cytotoxic T lymphocytes. Cell toxic T cells. So they kill other cell types. This particular cell can be distinguished from the inflammatory T cells by a different molecule that it produces called CD8. And CD4 and CD8 are the two main markers that distinguish two major subsets of T cells. There are a lot of other markers that will then further subset and subset and subset these cells into very functionally defined populations that we won't talk about at all here today. Anyway, these CTLs are able to kill virally infected host cells, eliminating the opportunity for virus to spread. It's a very important mechanism for controlling viral infection. Oh yeah, one more time I want to remind you to note the absence of B cells in CMI. So there's no antibody involved in any of this. This is all T cell mediated, and with the collaboration of other cells like cells of the innate immune system and the dendritic cells that provide antigen presentation and all of that stuff. So, but T cells are the main players, and there are no B cells around doing much of anything. Okay, so just to remind you before we move into another little topic I wanted to discuss today, or during this segment, is that we have layers of host defense. And the notion is, as we said from the very beginning, that there are microbial pathogens that are out to get you. Here you are down here, you're the host. And that pathogen has to get through these various layers of defense in order to get to the host. The layers are very efficient, effective at doing what they do. In the beginning, the initial layers are not very sophisticated, but they still perform a pretty vital function nonetheless. We have those physical barriers to infection, like the skin and mucous membranes serve as just walls between us and our outer environment. And then physiological barriers involving things that our own body does, that it's just there. The mechanism is there. You don't have to create the mechanism. There's a fever response, and there are nutrients that can limit the growth and survival of certain pathogens, and oxygen requirements and so forth, as we discussed in the first segment of this series. And if that doesn't work, if the bug breaches, gets through the physical barriers, and adapts somehow to some of these physiological barriers, so they don't care if it gets a little warmer. They're able to use whatever nutrients might be there, even if they're not the nutrients that they want. They might be able to adapt to the presence of oxygen, which they didn't really like at first. So they can adapt to these things in a variety of ways. They can mutate. They can do all kinds of things. In any event, they might be able to get through that physiological barrier. So this is all, it's a battle. I mean, we have the wall, the outer wall, and then we have some of the inner walls of, say, your castle that this microorganism is trying to attack. And then if the physiological barriers don't work, we have inflammation that might help. We have the granulocytes and macrophages mainly doing their phagocytic functions, which is great, except that they can only ingest so much at a time. And if the microorganism replicates and spreads faster than these cells of the myeloid cell lineage, these granulocytes and macrophages, if the infection progresses faster than the phagocytosis can occur, then we have trouble. But the idea is that if we just slow things down enough, get those granulocytes and macrophages to interfere with the spread within the body of a particular infection, then we give ourselves a little time, we buy ourselves some time. Because during all of this, which might be happening within the first few days after the infection has occurred, there are other cells that are beginning to become prominent, cells that we consider to be part of the adaptive immune system. These are mainly lymphocytes, the B cells and T cells that provide us with antibody, that provide us with CMI and all of that stuff. And so most of the time, that's enough. And these lymphocytes will take care of business. And so the infection was slowed down by these physical barriers, these innate barriers to infection, and then the infection is wiped out by the weapons that are generated during the adaptive immune response. Most of the time. Sometimes, though, our immune system just doesn't have what it takes, and the pathogen might, in fact, reach the host. And when it does, we're goners. And I mentioned early on this notion of there being sort of a co-evolutionary race that's always happening, where the pathogen over time will evolve new ways to get through these barriers, and the host in the meantime has to respond genetically, for the most part, to provide new weapons for defending itself. And it's a back and forth sort of battle. The pathogen will do something that the host will then respond to, and then the host will do something that the pathogen then will respond to in a genetic sense. So that back and forth, until finally somebody will win, the pathogen or us. Hopefully it'll be us. Only time will tell. But what I wanted, that kind of leads me into this last topic that I want to discuss with you in this third segment. The immune system is not infallible. Everything that we've been describing is the optimal sort of scenarios that we would like to think go on all the time, where we're always the winners. The immune system never does anything wrong. But the fact is, the immune system does do things wrong more times than we even know sometimes. And so we have this sort of war going on. This is the way I look at it. I keep talking about weapons of the immune system and strategies and adaptations and all this other stuff. I really think about this being a war between the immune system, our immune system, and the pathogens that are trying to defeat us. So the pathogens are invading our castle, and so we have these various layers of defense keeping them out of our castle. But sometimes things go wrong, and so I just wanted to discuss a few of those things. So, as in any battle, collateral damage may be observed. So I continue on with this analogy. And this is often a big surprise to some people. As I said, we usually think about the immune system as being nothing but good. It's protecting us from infection, so how could that be a bad thing? But it can be a bad thing. So like in any battle, potent force is often a two-edged sword. And so I drew this sword. I hope you all appreciate the amount of time it took me to create this sword. Anyway, so this two-edged sword, you can see there's two edges. Here's one over here, and that edge is the good edge. And it's the edge that represents the successful immunity that we can generate against viruses or bacteria or fungi, parasites, any of those pathogens that are out to get us. So that's the good edge. But then there's a bad edge too on the other side. And on the other side, we have the edge that can create certain immunopathological scenarios. So any of the inflammatory diseases, any of those diseases that have itis at the end, all the itis diseases, hepatitis, meningitis, and so forth, those are all diseases that are caused by the immune system kind of going off and doing something we wish it wouldn't do. So that's one way that the immune system can go wrong. We also have instances where there's excessive or inappropriate activities produced as the result of immune responses, like in some of the allergies that we know about. In fact, we're going to talk a little bit about that in a minute. The inappropriateness of these responses is only that there's too much of it, and it kind of spills over, the destructive effects spill over onto host tissues. So that's another possibility. And then there's certain diseases that are caused because the immune system fails to continue distinguishing between self and non-self. I think somewhere in the second segment, I can't remember when exactly, I made the point that we have all of this specific stuff going on where the immune system can recognize and then attack various antigens, very specific way. Lots of different destructive activities. But during all of that, for the most part, under normal healthy circumstances, all of that mass destruction occurs without hurting our own tissues. It's really phenomenal. And there are all kinds of very elaborate mechanisms that have been discovered that are responsible for preventing the immune system from attacking ourselves. And that's great, but it doesn't always work. And there are certain instances where the immune system fails to distinguish between self and non-self. In other words, where the immune system sees erythrocytes floating around in our bloodstream, and for some reason decides those erythrocytes are foreign and must be eliminated. And that would be a bad thing. Or numerous other tissues, maybe a muscle or nerves or liver or whatever. There are all kinds of examples of autoimmune diseases. Some of the common ones like lupus and rheumatoid arthritis and myasthenia gravis and multiple sclerosis. These are all autoimmune diseases where the immune system has lost the ability to recognize the fact that these self tissues are to be left alone. So that's autoimmune diseases and autoimmunity. It's I could lecture on that for hours and hours, and we just don't have that much time. But those are other ways that the immune system can go wrong. So these are all part of that bad edge of the sword. And the way I look at this sometimes is that that when we have an infection and we start creating immunity or generating immunity against that infection, that there's a wide range of possible outcomes, and that's what this diagram is supposed to indicate. So the immune system starts off at a very low level. When the infection first starts, the amount of immunity that exists at that point is so low that it's totally ineffective and really is not able to do much in the way of eliminating the infection. And under some circumstances, it may even stay at that level. I mean, in certain immunodeficiency diseases, for instance, you may never generate any immunity at all. So we have that end of the spectrum where we have ineffective host defenses. And then as the infection progresses under normal healthy circumstances, we generate more and more immunity, and so we get more and more effective at eliminating the infection. But then if we continue to intensify these immune responses, sometimes, as I mentioned a second ago, the destructive effects of those responses might spill over onto host tissues. And so we get a little bit of collateral damage where the tissues are damaged, the tissues are destroyed, but not completely. They're just, they're minor, well, I don't want to call them inconveniences because they can become life-threatening under the wrong circumstances. But for instance, when you get rid of a viral infection, like the flu, when you get the flu and you eventually kick out the virus at the end of it all, you do that because there's so much inflammation directed at the progressing infection that you're killing lots and lots and lots and lots of host cells that have the flu virus in them. And that's great, get rid of the flu virus. But at the same time, you're getting rid of your own cells too. So that's the collateral damage that I mean. Most of us, though, will recover from that, and that's fine. But in some individuals, that level of destruction may reach very significant and pathological levels, and you can generate severe pathology and creating acute or chronic inflammatory disease and so forth that can become, life-threatening. So there's a wide range of possibilities. And left unregulated, this would be more common than it is right now. But there are a lot of cell types in the immune system that are designed specifically to shut this down before it gets too bad. So that's another area of discussion that we could have another time. Okay, so let's just move into one area of where the immune system messes up a little bit. This is the area of hypersensitivity and allergy. So I'm going to just say a few words. It's just a few slides. It shouldn't take too long, describing some diseases that are caused by the immune system responding too well, and that its destructive effects spill over onto host tissues and create pathology. So I love this cartoon with regard to allergy. Bit of a bummer, really. You've got a nut allergy, and there's a little squirrel there. Okay, you get it. Okay. All right, so hypersensitivity diseases. A little definition of these diseases, and it is a collection of diseases. These are diseases in which there are exaggerated manifestations of normal host responses. Remember, when a word is colored like this, like in blue, means I really want you to remember that word. So please remember it. So exaggerated. So these are immune responses that occur using pretty much the same principles and mechanisms that we've described already for normal immune responses in the last two segments of this series, but they are in some way or another exaggerated. So they're bigger than they should be, hypersensitivities. So exaggerated manifestations of normal immune responses that results from exposure to antigens. Sometimes we call them allergens to signify that they're connected with allergy. In atopic individuals, atopic individuals are, that's a word that the geneticists came up with to indicate that someone is genetically predisposed to getting an allergy. So if someone's atopic, means that they get a lot of allergies. You probably know somebody like that. They're allergic to everything. So those are atopic individuals, creating a sensitized state, and then subsequent exposure to the same antigen elicits symptoms of immunopathological reactions. And we're going to show one particular example of that in a second. Okay, so there are four major classes of hypersensitivity. We don't have time to talk about all of them. We're going to talk a little bit more about one of them and leave the other three for another day. Type 1, 2, 3, and 4. Type 1 hypersensitivity, which is the one that we're going to talk about in a little more detail in a minute, often referred to as immediate hypersensitivity because of how fast it happens. It's associated with the release within just a few minutes of pharmacologically active agents from mast cells or basophils, sensitized with IgE antibodies. So this is a type of hypersensitivity that is dependent on antibody, but one particular class of antibody. Remember, antibodies have different heavy chains that give them different class designations. And this antibody, IgE, has an epsilon heavy chain, so it makes it IgE. So that IgE antibody is involved in all this, and we'll see how in a minute. This kind of hypersensitivity is associated with a lot of common things that insect bite, with that mosquito bite that you get, the food allergies that we talk about, like peanut allergies and so forth, certain drug allergies. So there are a lot of different things that can trigger type 1 hypersensitivity, immediate hypersensitivity. Type 2 hypersensitivity is sometimes referred to as cytotoxic antibody hypersensitivity. Remember, I said before, cytotoxic means toxic cell killing. So this is a type of hypersensitivity where antibodies are produced, primarily IgG antibodies, that will bind to particular tissues, cells or tissues, and then destroy them. There are various examples of type 2 hypersensitivities, and many of them are the classic autoimmune diseases, like lupus, like rheumatoid arthritis, and so forth. So that's type 2 hypersensitivity. Now, if you were going to distinguish between type 1 and type 2, or if I was anyway, I would start with the fact that they, although they both rely on antibody, one is associated with IgE antibody and the other is associated with IgG antibody, sometimes IgM as well, but IgG mostly. So that's a good way to distinguish them. Okay, the other thing that would distinguish them is that type 1 hypersensitivity is associated with release of these pharmacologically active agents from mast cells and basophils, whereas IgG antibody collaborates with complement. Remember complement, all those complement components that can produce, C3b and C5a and the MAC attack and all of that stuff. If you don't remember that, you could go back to the first segment of this series and think about it a little more. But anyway, complement together with antibody is a really potent destructive force. And that is the main force that destroys or damages or compromises whatever the tissue is that's being messed with. Type 3 hypersensitivity is sometimes called immune complex disease because there's a formation of large molecular complexes that end up depositing themselves in sensitive tissues and then creating destruction. Okay, so let me go back and kind of sort that out. In this particular kind of situation, what we have is the production of IgG antibody. Okay, so this doesn't distinguish type 3 from type 2 because both are associated with IgG antibody. But in the case of the type 2 hypersensitivity, if we go back here for a second, this antibody is specific for particular tissues. So the antibody will be specific and able to target nerve cells or liver cells or muscle cells or red blood cells, whatever it is. It's antibody with the antigen combining sites, the antigen binding sites with biochemical complementarity that recognizes and binds to particular molecules associated with particular tissues. So it's very tissue specific. So type 2 hypersensitivity is tissue specific. Now, if we go back to type 3 hypersensitivity, there's IgG antibody too also, but in this case, the IgG antibody is not specific for binding to the tissue that ends up getting destroyed. Instead, the IgG antibody is specific for something else, some third-party antigen that's circulating very often in the bloodstream, so that the IgG combines with that antigen in the circulation. Remember, the antigen is not part of the tissue that's ultimately going to get destroyed. It's something altogether different. An IgG antibody binds to that something else, and they form complexes together. In fact, and the complexes get rather large, and when they get rather large, they end up getting stuck in particular tissues. They get stuck in certain filtration tissues, for instance, like in the kidneys. So immune complex disease or type 3 hypersensitivity is often associated with kidney damage. They also get stuck in capillary beds, where the blood vessels get really small in diameter. The complexes are so large, they're virtually, they are macromolecular. If you take somebody's blood and look at the serum from that blood, if those complexes in type 3 hypersensitivity are present, you hold up the serum, which is usually clear. It's got a color to it. It's kind of an amber color, but it's usually clear. You can look right through it. But in an individual that's got these complexes circulating, it's cloudy. It's so cloudy because of these complexes that now have formed. Those complexes get stuck in places where you really don't want them to be. And then they activate complement as well. So similar to type 2 hypersensitivity, in type 3 hypersensitivity, the complement cascade is activated. The main difference between type 2 and type 3 is that in type 2, the antibody is specific for the tissue that gets destroyed. In type 3, the antibody is not specific for the tissue that gets destroyed. It's specific for some third-party thing, whatever. And in both, complement is activated and is the destructive force that causes the pathology. So there are similarities and there are differences between type 2 and type 3 hypersensitivity. Type 3 hypersensitivity, there are various examples of that as well. Serum sickness is probably the first and the main one that's always discussed, where antiserum from some source other than human is administered to a human being. You might think that's kind of crazy, but a lot of the antitoxins, for instance, somebody gets bit by a snake, and you have to neutralize the venom of the snake before it kills the person that got bit. We often will inject a big bolus of serum with antibodies in it, an antiserum, that will track down the venom and destroy it before it can destroy us. The catch to all this is that the serum often comes from, the antiserum often comes from horses and other large animals where we can generate a lot of antiserum. And so what you're really doing is you're injecting a big whopping dose of horse proteins into an individual, which the individual's immune system will respond against and create antibodies against it. So the antibodies from the host start to combine with the antigens from the horse, and you make these complexes, and then you get kidney failure and all kinds of weird stuff if you don't manage things just right. Okay, then type 4 hypersensitivity is the easiest one to distinguish from the other three because there's no antibody involved at all. It's a cell-mediated hypersensitivity. Most of the action is connected with T cells and macrophages that destroy antigen-coded cells. There's contact dermatitis, like poison ivy, and some other sorts of allergies like that are examples of type 4 hypersensitivity. Anyway, we need to move on. All right, so we're going to discuss a few more details concerning the type 1 hypersensitivity. This is the IgE-mediated hypersensitivity. So here we have this is the mucous membranes in the lungs, say, and imagine for a second that somebody moves to San Antonio from up north. This person came from Chicago or someplace where there's no mountain cedar pollen at all, and so they move down here and they hear all these horrible stories about people having these major horrible allergies against mountain cedar pollen, and it's kind of scary. And they start, this guy that just moved down here starts wondering if he even should have moved down here and all of that. So, all right, anyway, so mountain cedar pollen season comes, and there's tons of this pollen in the air, and you know that it's here because any of your friends that have allergies against mountain cedar, start wheezing and sneezing and eyes tearing up and runny noses and all of that sort of stuff. Meanwhile, this guy that just got here, nothing's happening. You have all this mountain cedar antigen basically floating around that he's inhaling right along with everybody else, but no symptoms. And he thinks, aha, I'm one of those lucky ones that's not allergic to mountain cedar pollen, and he's feeling really good about that. But what might be happening is here's the antigen, and the antigen crosses these mucosal barriers and reaches immune tissues, lymphoid tissues, where it gets taken up by antigen-presenting cells, those dendritic cells that have to present antigen to T cells. And then those antigens are in fact presented to T cells, a particular subset of T cells that we call Th2 cells. Don't worry about why it's called Th2, but that's what it does. The main thing the Th2 cells can do, though, is produce a whole bunch of a particular cytokine. Remember cytokines? We discussed interferon gamma a few minutes ago, one of the inflammatory cytokines. Well, there's a whole big whopping, huge list of different cytokines that different cells can make. Another one is called interleukin 4, or IL-4. And Th2 T cells are capable of making a whole bunch of IL-4. The reason that that's important is that IL-4 is a really important stimulus for activating B cells with the ability to make IgE antibody. IgE antibody. And if there's more IL-4 around than there should be, most of the antibody that's going to be produced against this original antigen up here, which remember it's mountain cedar pollen antigen, most of the B cells that are activated against that antigen are going to be producing IgE antibody. IgE antibody. Now, in other individuals where the level of IL-4 production is much lower, which is most of us, there might be a little bit of IgE produced, but the vast majority of antibody that's produced against that mountain cedar antigen is going to be IgG antibody, which cannot do what I'm about to describe. So here's the IgE antibody, and this happens. Remember, this is just an immune response, like every other immune response that we've talked about. We showed the kinetics of the production of antibody to be a 1 to 4 week process or so. So over those period of weeks, there's a bunch of IgE that's being produced. To this point, though, this new guy that just moved to town is watching all of his buddies or work colleagues or whoever really, really suffering from mountain cedar allergy, but he's not. What he doesn't know is that during those weeks that he's kind of snickering at all the suffering that's going on among all those other people, he's making this IgE antibody. No symptoms yet, nothing. I mean, the IgE itself can't hurt you, but the IgE is in fact being produced. And then what happens is that a lot of that IgE will get anchored to the surface of a very special set of cells, mainly mast cells, sometimes basophils. These are leukocytes that we really didn't spend much time on earlier, but they're leukocytes that tend to live in areas right under the skin. They like to live along the mucous linings of particular tissues, like in the lungs and so forth. And so these mast cells have very special molecules on their surface shown in red. They're called Fc receptors. Now they're called Fc receptors because if you remember your antibody structure, antibodies have antigen binding regions associated with them, but they also have this sort of long tail that's referred to by the structural biologists as the Fc region. Call it the Fc region because it crystallizes really easy. So that's what the C stands for. It doesn't matter really, but anyway, so the Fc region of the antibody is the opposite end from the end that has the antigen binding sites. So here they are, stuck into the Fc receptor by their Fc region, and their antigen binding sites are sticking outward. Yeah, outward, right. And so during these weeks that the response is going on, these mast cells are getting loaded up and loaded up and loaded up with these IgE antibodies that are all specific for mountain cedar pollen antigen. Still, no symptoms. So this guy thinks he's in the clear. So now mountain cedar, this cloud of mountain cedar that came into town, rolls back out of town, and mountain cedar pollen season is over, and no symptoms. So the guy is really happy. But then next year, mountain cedar pollen season comes again, as it always does. And what we find is that there's a new source of antigenic stimulus that's come into town that can cross into this guy's lungs. So he starts inhaling all this stuff. And what he doesn't know is that all these mast cells with Ig antibody against mountain cedar pollen is just sitting there waiting in his lungs. So the antigen comes in and binds to the IgE antibody because the antigen binding sites are facing outward, and you end up triggering the elicitation of symptoms. So this original activity where the antibody is produced and the mast cells are bound by IgE antibody is often referred to as the sensitization phase in this allergic scenario. So this person is said to be sensitized to this allergen. Doesn't mean that there's symptoms, it just means he's sensitized. So you start with sensitization over here on the left, and now we're going to get elicitation of symptoms on the right when a second exposure occurs. So the antigen comes into town, passes into his lungs, and ends up binding to these IgE antibodies. Now, if you remember, I made a big deal out of the fact that antibodies have two binding sites that are identical in their specificity for reacting with the exact same antigen. The molecules, the antibody molecules, are said to be bivalent, two valencies for binding antigens. So what's able to happen is that one antibody on the surface of this mast cell can bind to this antigen, and then another antibody right next to the first antibody can also bind to that antibody, that antigen, and you get this sort of bridging effect, cross-linking they call it. So the antigen cross-links these receptors, and the physical result of dragging these receptors, these IgE molecules, closer together is to trigger signals that go into the mast cell. And there's a whole long sequence of biochemical events that occur that we're not going to talk about today. But the end result of that signaling is that there is a process triggered that we refer to as degranulation. So these mast cells have bunches of these sort of big, fat, juicy granules. And inside the granules are a lot of pharmacologically active mediators that just kind of sit in the mast cells under resting circumstances. But if you start cross-linking IgE on the surface of those mast cells and drag these Fc receptors together, it drives signals into the mast cells that stimulates the release of the contents of these granules. And you can see all these little dots. These are all these pharmacologically active mediators that are going to go out into your body and make you miserable. One last thing, these Fc receptors that I mentioned are sometimes further defined as being Fc epsilon receptors. See the epsilon there? So that indicates that these are specific for binding to Ig or antibody molecules that have the epsilon heavy chain in them. There are other scenarios that we could talk about, not today, where there are Fc receptors that are specific for the gamma heavy chain as well. So they would be Fc gamma receptors. Anyway, these are Fc epsilon receptors. And the point of all of this is that within just a few minutes, these cells are degranulated. You know, you inhale this pollen, you get this binding. It takes a few minutes for the antigen to get to these mast cells. But once it gets there, the mast cells don't have to do much of anything to release the contents of their granules. So within a few minutes, 15 minutes, 20 minutes max, you start getting really high levels of these mediators in the bloodstream that can then go on to cause pathology. All right, so what are these mediators? What are some examples of these mediators and what do they do? The star of the show in general, when you talk about type 1 hypersensitivity, is histamine. When histamine is released, the cells that it target will result in increased diameter of blood vessels. So you get vasodilation. You also get increased permeability of the vasculature. So remember, we talked about increased vascular permeability when we were talking about inflammation and host defenses by the innate immune system. Well, increased vascular permeability is a good thing to a certain extent if you want to get neutrophils and macrophage or monocytes or whatever out of the bloodstream to get to an infection. A little bit of increased vascular permeability is okay. But under these circumstances, there is excessive increased vascular permeability. So the vasculature becomes very leaky. So we have increased diameter of the blood vessels and increased leakiness of the blood vessels. And then in addition to that, not directly connected with it, but ultimately connected with it, is that histamine will stimulate smooth muscles to contract. Now, these aren't the voluntary muscles that we usually, the skeletal muscles that we usually think about. These are muscles that surround, like the lungs. The lungs are surrounded by smooth muscles, helps the lungs expand and contract and so forth. And there's smooth muscles all over the body. In any event, histamine will stimulate these muscles to contract. So what you end up with is kind of fluids leaking out of the vasculature, starting to fill up the lungs, and then smooth muscles that surround the lungs contracting and kind of pressing down on the lungs, making it more difficult to expand the lungs. So that's part of what histamine does. And this is, histamine is that's we run to the Walgreens or CVS or wherever to get our antihistamines. We're trying to counteract that, and we'll talk a little more about that in a second. Anyway, a second uh, uh, major mediator, actually pair of mediators, are the leukotrienes and prostaglandins. These do some of the same sorts of things as histamine does. They increase vascular permeability, so increase the leakiness of the vasculature. They also stimulate smooth muscle contraction, so so that activity is compounded over what histamine does. But then they have this extra added attraction, which is that they stimulate high levels of mucus secretion. So now you have mucus being secreted and pumped into the lungs, along with any of the other fluids that might be collecting because of the increased vascular permeability. And now you got your lungs filling up pretty good with fluid. So you have the fluids filling up the lungs, and you have smooth muscles, kind of bearing down on the lungs, keeping them from expanding. You can imagine that this is going to make it harder to breathe. And that's one of the main symptoms of type 1 hypersensitivity is labored breathing. And in fact, if it gets too bad, you could have full time or big time respiratory collapse. There are a couple of other things that we find released from these mast cells, like certain degradative proteases. These are proteins, these are enzymes that'll break down the integrity of the tissue and allow the other mediators to get even deeper into them. So degradative proteases. Certain chemokines are really important. These chemokines are, if you remember from the very first couple of slides in part 1 of this series, chemokines are small molecular weight proteins that attract cells, chemokines. These are chemoattractants that attract certain cells of the innate and adaptive immune system, for that matter. So we have two main players. One is eosinophil chemoattractant factor, chemotactic factor, and the other one is neutrophil chemotactic factor. So the neutrophil chemotactic factor attracts neutrophils, obviously, but eosinophil chemotactic factor is really important in this scenario for a very practical reason, which is that eosinophils usually are found in extremely low numbers. If you look in the blood, if you look in mucus, if you have a cold or something and you're blowing your nose and you look in the mucus, there might be a few eosinophils in there, but eosinophils usually are very shy and they stay out of the picture. But when you degranulate a whole bunch of mast cells and you stimulate or you release a whole lot of eosinophil chemotactic factor, you get a parade, a tsunami of eosinophils coming out of the bone marrow where they're made. Remember, those hematopoietic stem cells in there make all the cells of the blood. Well, in this case, this chemotactic factor attracts all of these eosinophils that are being made in the bone marrow and increases their production, in fact. And we end up with extremely elevated levels of eosinophils in the blood and in mucus secretions. And it ends up being so significantly elevated that it becomes almost diagnostic all by itself for type 1 hypersensitivity. So somebody, while they're suffering through this mountain cedar problem, they blow their nose or whatever, if you took that mucus and you examined it under a microscope, it would just be cram-packed with eosinophils. So eosinophilia is highly diagnostic of type 1 hypersensitivity. Then there's certain cytokines that activate leukocytes to do a variety of inflammatory things that also aggravate the situation. Type 1 hypersensitivity can have its effects in a lot of different places in the body. The most obvious and probably the least consequential are type 1 hypersensitivities that involved the skin. In fact, it was, I think, probably skin responses that were first described when people discovered type 1 hypersensitivity. And they described this so-called wheel and flare reaction. Here's somebody's forearm. And what this is really is a skin test for a particular allergen. I don't know what the allergen is, but we're trying to see whether or not this person's allergic to it. And so what they do is they inject small volumes of it right under the skin. And there's 4 injection sites. And at each injection site, there is increasing, as you go up the arm here, there's increasing concentrations of the allergen. So we see within 20 minutes, as you can see down here, 20 minutes, within 20 minutes, we're seeing this lesion created. At first, it's sort of small and subtle, but as you increase the dose of the allergen, it gets quite severe. This middle section, this little dot, is filled with mainly fluid. If you touched it, it would be squishy. Squishy is that, it's a highly clinical term, I know, but anyway, it's filled with fluid. And then you get a little bit of erythema that's observed surrounding the primary lesion. And as you go up and increase the doses of the allergen, you see that the flare out from the primary lesion gets bigger and bigger and bigger. So this is the wheel and flare reaction. And this is indicative of somebody that's allergic to whatever this allergen is that we're testing. So insect bites, a lot of, if you get bit by a mosquito, that's a type 1 hypersensitivity, and you're going to see a wheel and flare reaction depending on how long the mosquito sat on your skin sucking your blood before you swatted it away, because the amount of allergen will be related to the size of the mosquito bite, sign of the mosquito bite that you see. A lot of the food allergies that we hear all about, peanut allergies, shellfish allergies, and so forth, these are type 1 hypersensitivities as well. The trouble, though, is that the reaction is not occurring on the skin where it doesn't do too much damage. Instead, it's happening along the intestinal tract where you do damage to the tissues of the intestines, but also stimulate a lot of other activities that result in fluid gushing into the intestinal tract and creating, causing diarrhea and cramping because of all the smooth muscles that are being stimulated to contract and so forth. So gastrointestinal sites of reactivity are common. And then there's the lungs. The scenario that we were just describing can be very subtle, relatively mild, or it could turn into, which we would refer to as allergic rhinitis, or it could turn into allergic asthma. And that can become quite severe and potentially even life-threatening under the wrong circumstances. And then finally, systemic type 1 hypersensitivity is referred to as anaphylaxis. And remembering that all of these mediators that we had described a few minutes ago end up changing the way blood vessels transport blood. We end up with a drop in blood pressure that can't be fixed by administering fluids, and you have a patient going into shock. And so we would refer to that as anaphylactic shock, which of course is life-threatening. Okay, so to finish this up, I brought a little case with me. Under normal circumstances, we'd be face-to-face and we'd have a good time, back and forth trying to figure this out. But I'm going to do the best I can to just kind of describe it and give you some additional insights into all of this best I can. So this is a 22-year-old woman. And we look at her, and right off the bat, we can kind of see that she's a bit bloated, swollen up. Something's going on here. We also notice when we look at her skin that up close especially, we see this sort of lacy looking rash. So something's going on in her skin, and And my first guess would be that she's demonstrating a good case of hives, urticaria. Okay, so she's outside in her backyard. She gets stung by a bee, and then this is what happens to her. She swells up. She shows this rash, and all of this happens within a few minutes. Working in her garden all summer had resulted in several stings prior to this one. In just minutes after being stung, she noticed an itching sensation and abdominal cramping. Soon she began to feel faint and acutely short of breath. Her pulse was rapid. Her lips and eyelids were swollen. So this thing is going on. Um, she'd experienced similar episodes before, which makes her not so bright. I mean, what was she doing out in the garden again if this had happened before? Anyway, she's out there and it's happening. The woman in the past had administered intramuscular injections of epinephrine, the famous EpiPen, and intravenous injection of a mixture of hydrocortisone and chlorpheniramine, which is a histamine receptor antagonist. And usually, and the last time, and then this time too, I suppose, injecting these mediators um, uh, took her back away from the edge of the cliff. Her symptoms subsided. When she visited her local ER for examination, an ELISA assay was done that verified that she had in fact just demonstrated a type 1 hypersensitivity caused by the presence of bee venom reactive IgE. So type 1 hypersensitivity, clearly type 1 hypersensitivity, clearly not very smart that she's out there in the garden again. Anyway, so I have a few questions that I would ask you, and I'll give you a 2 or 3 second pause for you to kind of think of what you think the answer is, and then I'll give you the answer. So what's the significance of the lady's previous history of bad luck with bee stings? Okay, as we mentioned before, there are two major phases in type 1 hypersensitivity. There's a sensitization phase, and then there's a symptom elicitation phase. So what this indicates is that this lady was sensitized and at some previous time. So she's been bit before by a bee, which stimulated anti-bee venom responses that were dominated by IgE antibody that bound to the FC receptors on mast cells, and then waited. Those mast cells waited. Those cells will wait for a very long time, sometimes years. They're very stable and they stay coated with that IgE antibody. Well, at that point, when she was stung the first time, the very first time she was ever stung, she probably didn't see any symptoms at all, but she was sensitized. The second exposure to that allergen, to that bee venom, is what did it and triggered the elicitation of symptoms that eventually resulted in the pathology that we observed. Time of onset of the symptoms, what's significant about that? Well, it happened almost right away. You could even say immediately. So this is an example of immediate hypersensitivity, type 1 hypersensitivity. Why'd she feel faint? Why did she feel faint? Okay, so as I think this through, I know that a lot of these mediators like histamine and leukotrienes and so forth, they will increase vascular permeability. They will also increase the diameter of blood vessels, so we get vasodilation. So what is that going to do to the blood pressure demonstrated by this person? So the diameter expands, so it takes less force to get the fluid through the blood vessels, so that's less blood pressure. And also the vessels are leaky now. So even though you're pushing forward to get this fluid through, some of it's going off sideways, and so blood pressure overall is going to be affected significantly. So blood pressure has dropped, and so what that means is that the amount of blood that reaches her brain is going to be significantly lowered. So less blood flow to the brain. Less blood flow to the brain means less oxygen to the brain. Less oxygen to the brain is detrimental to the function and health of the brain, and feeling faint is one of the first signs that the brain is feeling sick. And why does she? Why is she short of breath? Why is she short of breath? Well, as we had mentioned before, uh, during this scenario, one of the things that's happening is that her lungs are filling up with mucus that's stimulated by the presence of the leukotrienes mainly. Lots of mucus filling up her lungs, so she's got a lot of fluid in her lungs. And in addition to that, we have histamine and the leukotrienes and the prostaglandins and so forth. They stimulate smooth muscle contractions, so we're contracting down on the lungs. Those muscles are kind of becoming rigid because they're contracted, and it makes it really hard to expand the lungs. So it makes it hard to breathe. And that combined with the fact that the lungs are filled up with fluid, making oxygen exchange very difficult. Uh, she's gasping for breath. She's just out of she's trying to get oxygen in there as best she can, and so she's going to have very labored breathing. So she's short of breath. The drugs that were administered, there was an antihistamine that most of the time the antihistamines are not drugs that actually destroy the histamine, but instead interfere with the receptor on tissues for histamine. So we have histamine receptor blocking by these histamine antihistamine inhibitors. We also had an anti-inflammatory to try and keep things a little bit more under control. And then finally, the significance of high levels of IgE. Well, that's the main bad guy. That's what triggers this whole thing off. IgE antibody is produced very prominently in atopic individuals that demonstrate this kind of allergic response. So IgE antibody will bind to mast cells, and then later on creating a sensitized state, and then later on the allergen will come along, the antigen will come along and bind the IgE, trigger those mast cells to degranulate. None of this happens if there isn't a lot of IgE around. Most of the rest of us that respond against these mountain cedar allergens and so forth, we make IgG antibodies that will not associate with mast cells to any great extent. And so we don't demonstrate the same symptoms as somebody that makes a lot of IgE. So that's pretty significant. And that's all we have. So we've gone through these three segments discussing innate immunity, adaptive immunity. We talked a little bit about where the immune system might go wrong. There's so much more we could talk about in terms of how the immune system goes wrong, but we just don't have the time. And I hope this helps clarify some things for you, or fill some gaps, or maybe even stimulate some new thought about how the immune system works and how it defends us from infection. And I really appreciate your attention, and I really appreciate your working with me to deal with this new reality, teaching reality, with the need to do more remote teaching. We're getting through it. We're all going to get through it, and I wish you a lot of health and safety and happiness. So thanks a lot. Bye.